Hello everyone. Today we're going to talk about hemostasis, and I'm going to explain it in the way that helped me understand it. As you guys know, this is a first year topic, and it's definitely not the easiest thing to wrap your head around at first. So I'm going to go step by step and explain it in a way that made sense to me. To start off, I think it's really important to have a clear definition of what hemostasis actually is. Before we dive into the details, hemostasis is the prevention of blood loss whenever a blood vessel is severed or ruptured. Hemostasis happens in stages, and we're going to go through those stages in order. First, we have vascular constriction, then formation of a platelet plug, and then formation of a blood clot as a result of blood coagulation. Together, these make up the hemostatic response. After that, there's also fibrous tissue growth, which permanently repairs and closes the vessel. We're only going to mention that briefly because the main focus of this video is vascular constriction, platelet plug formation, and blood coagulation. If we look at this visually, it's a bit easier to understand. First, the vessel constricts, meaning the diameter becomes smaller. Then platelets gather at the site of injury to form a platelet plug. After that, a blood clot forms through the coagulation cascade. Finally, fibrous tissue is involved in repairing the vessel wall and completing wound healing. So starting with vascular constriction, which is also called vascular spasm. These two terms mean the same thing. This is the first response to blood vessel injury. What happens here is that the vessel lumen narrows, so less blood is able to escape. Remember, the goal of hemostasis is to stop blood loss. The purpose of this vasoconstriction is to buy time for the next steps, platelet plug formation and blood coagulation. If you imagine a blood vessel, especially arteries and arterioles, they have a smooth muscle layer. When the vessel is damaged, this smooth muscle contracts. That contraction is what we call a vascular spasm. As a result, the vessel diameter decreases, blood flow drops immediately, and blood loss is reduced straight away. This doesn't happen randomly. There are specific mechanisms that cause vascular constriction, and we'll go through those now. The first mechanism is local myogenic spasm. This is a contraction that originates directly from the smooth muscle itself. Damage to the vessel wall directly stimulates the smooth muscle in that wall, and the muscle responds reflexively by contracting. The important thing here is that this does not require nerves or hormones. The smooth muscle essentially reacts instinctively to being injured. The second mechanism involves local autocoid factors. Autocoids are locally acting chemical substances released by injured tissue and activated platelets. They are not systemic like hormones. Instead, they act close to where they are released. A key autocoid to remember is thromboxane A2. Thromboxane A2 is released by activated platelets at the site of injury. It causes vasoconstriction and also promotes further platelet aggregation. This is especially important in small vessels because platelets are closer to the vessel wall, so the chemical signals have a stronger effect. The third mechanism is nervous reflexes. Injury causes pain, which stimulates pain receptors and sensory nerve endings. This generates nerve impulses that trigger vasoconstriction. These nerve impulses can also directly stimulate local myogenic contraction, reinforcing the vasoconstriction. So to summarize vascular constriction, it is the immediate first-line response to vessel injury. It reduces blood loss through local myogenic spasm, autocoid release, such as thromboxane A2, and nervous reflexes. The more severe the injury, the stronger the response. These mechanisms are more effective in smaller vessels, and they are sustained long enough to allow platelet plugging and coagulation to take place. Next, we move on to platelet plug formation, which happens after vasoconstriction. If the injury is very small, the vessel may be sealed by a platelet plug alone without needing a full blood clot. Many tiny injuries in blood vessels happen every day and are sealed this way without us noticing. Before explaining the plug itself, it's important to understand the platelet. Platelets, also called thrombocytes, are derived from megakaryocytes in the bone marrow. They have no nucleus, and they cannot reproduce. Inside the platelet, cytoplasm, are contractile proteins, actin, myosin, and thrombostatin, which is platelet-specific. These proteins allow platelets to change shape and contract after activation. This contraction pulls platelets tightly together, helping to seal the injury. Platelets also contain residual endoplasmic reticulum and Golgi apparatus, which support enzyme synthesis and calcium storage. Calcium is essential for platelet activation, aggregation, and multiple steps of the coagulation cascade. Platelets contain mitochondria, which produce ATP and ADP. ATP provides energy for contraction and shape change, while ADP is a powerful platelet activator. Platelets also synthesize prostaglandins, which are short-lived, locally acting signaling molecules. These promote vasoconstriction, platelet aggregation, and inflammation. In addition, platelets release growth factors that stimulate endothelial cells, smooth muscle cells, and fibroblasts. This promotes tissue repair and vessel healing, so platelets don't just stop bleeding, they also promote healing. Platelets have a glycoprotein coat, which prevents them from sticking to normal, intact endothelium, but allows them to adhere to damaged vessel walls. This prevents unwanted clot formation in healthy vessels. Now back to platelet plug formation. In a normal vessel, platelets circulate freely and do not stick to the vessel wall. This is because the endothelium is intact and collagen is not exposed. When a vessel is damaged, the endothelial layer breaks, exposing collagen underneath. Platelets cannot bind directly to collagen. They need help, and this is where von Willebrand factor comes in. Von Willebrand factor is a sticky plasma protein that binds to exposed collagen and simultaneously binds to platelet surface receptors. You can think of it like double-sided tape. One side sticks to collagen, the other to platelets. This anchors platelets to the injury site. Once platelets adhere, they become activated. Activation causes them to change shape from smooth discs into spiky cells with extensions called pseudopods. These increase surface area and improve adhesion to collagen and to other platelets. Activation also causes the contractile proteins to contract, pulling platelets tightly against the vessel wall. This contraction forces platelet granules to release their contents, including ADP and thromboxane A2. ADP activates nearby platelets, while thromboxane A2 causes vasoconstriction and further platelet aggregation. Platelets flowing past detect these signals and become activated even before contacting collagen. This creates a positive feedback loop. As more platelets stick together, a platelet plug forms and physically blocks the hole. This process is called primary hemostasis. Sometimes the platelet plug is not enough. High blood pressure, strong blood flow, or large injuries can cause the plug to break apart. In these cases, we need blood coagulation. Blood coagulation is also called secondary hemostasis. Normal blood contains both procoagulants, which promote clotting, and anticoagulants, which prevent clotting. In intact vessels, anticoagulants dominate. When a vessel is damaged, procoagulants become activated locally and override anticoagulants only at the injury site. Endothelial cells normally release prostacyclin and nitric oxide, which prevent platelet adhesion. When the endothelium is damaged, this protective effect is lost. Secondary hemostasis stabilizes the platelet plug by forming a fibrin clot. This is especially important when the platelet plug is loose or easily disrupted. The key step is formation of prothrombin activator, which acts like the on switch for clot formation. Prothrombin activator converts prothrombin into thrombin, and this step requires calcium. Thrombin is a key enzyme. It converts fibrinogen into fibrin. Fibrin monomers then polymerize into fibrin fibers, which are stabilized by factor 13A, also called fibrin stabilizing factor. This creates a strong, cross-linked fibrin mesh. The fibrin clot traps platelets, red blood cells, and plasma, and anchors the clot to the vessel wall. This transforms a fragile platelet plug into a stable clot. Coagulation occurs through two pathways, the extrinsic pathway and the intrinsic pathway, which converge at the common pathway. The extrinsic pathway is triggered by tissue damage outside the blood vessel. Damaged tissue releases tissue factor, also called factor 3. Tissue factor combines with factor 7A to form a complex that activates factor 10 into factor 10A. The intrinsic pathway is triggered by damage to the endothelial surface inside the vessel. Exposure of negatively charged collagen activates factor 12, which then activates factor 11, which activates factor 9. Factor 9A combines with factor 8A to activate factor X. Both pathways lead to formation of factor XA, which marks the start of the common pathway. Factor XA combines with factor VA to convert prothrombin into thrombin. Thrombin then converts fibrinogen into fibrin and activates factor 13, which cross-links fibrin to form a stable clot. Thrombin also amplifies the cascade by activating factors 5, 8, and 11. And that brings together the full process of hemostasis, vascular constriction, platelet plug formation, and coagulation working together to stop bleeding and allow vessel repair.